Here. So this is where we're starting. We have this double-stranded helix, this DNA, okay? And the first thing we're going to do is we're going to open up at a site called the replication fork or the replication bubble. So I'm just going to draw that. Somewhere along this DNA strand, we're going to have this site where an enzyme called helicase is going to attach to. So helicase, ACE, means it's an enzyme. If you guys remember, whenever we did nutrient stuff, we practiced those root words like ose. So ose is a sugar, ACE means enzyme. So we have an enzyme, helic has to do with helical or helices. So this helix, this double-stranded helix DNA, this helicase is an enzyme that works on this helix. So what it does is it unwinds it. And that's at this site. Where the helicase attaches is called the replication fork or the bubble. Alright, what I want to do next is take a look at this. So what this is two nucleotides. So if you remember, whenever we were going through those macromolecules, we talked about nucleic acids being made up of these monomers, nucleotides. So a nucleotide is made up of three things: a nitrogenous base, this is thymine, and that's adenine, but it's also made up of a ribose sugar, either deoxyribose or ribose. Since this is DNA, this is deoxyribose, and what that meant is it didn't have an oxygen here like ribose normally does, so we get rid of that oxygen and it's just a hydrogen. So be a hydrogen bound there. Go back and look at the videos on macromolecules if you want to get some more pictures on that. When we talked about how we connect nucleotides, how do we make a polymer or a long nucleic acid, we have to have this phosphate group make this bond. So this is called a phosphodiester bond. This is the bond in between. So whenever I'm looking at my ribose sugar, I know it's a pentose sugar, meaning 5 carbons. I have 1 carbon, 2 carbon, 3 carbon, 4 carbon, and 5 carbon. Now, whenever we add a nucleotide, we add from the 5 end to the 3 end. So this is, if you look at this, this is a 5 prime and this is a 3 prime. So when I'm making my new DNA strand, I'm going to be building from a 5 prime to a 3 prime. So we know complementary base pairing is going to put a T here, and this T would also have a ribose sugar, and we would have a phosphate, and boom, and we'd go like this. Phosphate, ribose sugar, and actually, yeah. So T and A would go together, A and T would go together. So all I wanted to establish right here, the structure of a nucleotide, how we're adding nucleotides from this 5 prime to 3 prime direction, and it's like if you're looking at your parent strand, it's going to add in the opposite direction that the 5 prime to 3 prime runs in your parent strand. And how can you identify this? Well, if you look at this very top, if we just drew a line and we said, what is the highest number here? Is it a 3 carbon or a 5 carbon? That's a 5 carbon. If I come down here and I just draw a line like this, boom. What's at the very bottom? A 5 carbon or a 3 carbon? This is a 3 carbon, so this means this is the 3 prime side. Alright, why is that important? Because we're going to talk about how nucleotides are added. But first I'm going to give you just, I'll write everything out, then we'll go back, add some pictures, and work through this thing. Again, this is anatomy and physiology, this isn't Bio 1, so you're going to be tested on this material one time. You definitely need to know how it works, but I'm going to try not to go into too much detail. So, we've already talked about this DNA unwinding because of this enzyme helicase, and that takes place at a replication bubble or a replication fork. Now we have exposed nucleotides, so these are going to serve as a template for complementary base pairing. All that means is that adenines are going to be paired with thymines and guanines are going to be paired with cytosines. So complementary base pairing. We're going to get A's going with T's and G's going with C's. So that's our template. We know that we have these exposed nucleotides. So these exposed nucleotides, something called a primer is going to bind to that. Okay, a primer is going to bind to it. And it's going to lay down some nucleotides. And then our friend DNA polymerase, and if you want to break up this word, this says ACE, that means it's an enzyme. And polymer, if you remember whenever we said that a protein is a polymer. Its monomers are are amino acids. If we talk about carbohydrates, carbohydrates are a polymer where glucose are monomers of these sugars. So here, if we want to get DNA polymerase, we want an enzyme that's going to make a polymer of DNA. So we want a bunch of nucleotides strung together. So this is the guy, DNA polymerase is the enzyme that adds nucleotides one at a time. How did we get from that name? We see ACE meaning enzyme, and we see DNA polymer. We want to make a big nucleic acid. So this is what's going to add our nucleotides. We'll talk about this lagging strand in just a sec, but remember this enzyme DNA ligase is what's going to connect those Okazaki fragments on the lagging strand. And then histone proteins, the things that DNA wrap around to get all condensed, are going to form, and then you'll have condensed DNA. So let me add some pictures. Helicase opening up this DNA, and so now we're going to have two DNA strands. And remember, we have a 5' end and a 3' end. All this means is that, if we are looking at our nucleotides, then on this end, I would have my 3' at the bottom, and it would go all the way up adding, and then finally we'd have at the top a 5' carbon. This other guy's going to run in the opposite direction, so we'll have 3' and 5'. Now again, these are parent strands. So we have to take these parent strands and add nucleotides only in the 5' to 3' direction. So let's say we're opening up this way. Boom, we're opening up that way, and let's work through that. My new daughter strands are going to be 5' to 3'. And we have a 5' to 3'. So, as this is opening this way, this is going to keep exposing a 5' to 3' end. So when you start with the 5, you're going to add your first nucleotide. We know that this parent strand, this end is 3', so we know this daughter strand, this will be a 5'. So we're going to add a 5, and we add from the 5 to 3 directions, so we'll add another one there, another nucleotide there, and another one there, all the way as we get down here. The problem with this top strand, called the lagging strand, is that we can only add in the 5 to 3' direction, so we can only add this way. Why is that problematic? Well, as my DNA is unwinding on this side, I keep exposing 5' ends, not 3' ends. So the problem with that is that I can't keep adding nucleotides this way. With this strand, I added a nucleotide, added a nucleotide, added a nucleotide. DNA polymerase is just going along this thing, adding nucleotides, because it's from a 5 to 3' end. This top strand, we can't do that. So what we have to do is we'll have to open up a little bit of DNA. So let's just say we go another, 100 base pairs that way, whatever. So now we're going to have from here to here, this 5' to 3' end. It's been open, DNA polymerase now has to act. So we can only add from 5 to 3', so we can only add in this way, but we're opening them this way. So what that does is we constantly have these different sections where we'll have gaps. We'll have this get filled in, but as it's been opening, DNA polymerase isn't working this way, it's working this way, so we get a gap here. So we get a gap here, and then we'd expose some more. So now we've moved down the replication bubble, we've moved down this DNA molecule. So now what we were just looking at, where that 5 to 3' end was exposed and we're adding this way, now we have all this space that needs to be connected. So we start from this 5' end and then we add nucleotides. And then it's going to keep opening up and we're going to have more 5' ends to add to. So then we add this way. So we have these little spaces right here and right here. We have spaces of emptiness that we need to put together to have this full DNA molecule. That's where DNA ligase is going to come in. So DNA ligase is going to connect these fragments on the lagging strand. Here's our lagging strand up here, and we know that we're adding from the 5' to 3' direction, so we keep having these different fragments. These fragments, this fragment that's had the DNA polymerase act on it, is called an Okazaki fragment. Okazaki. So DNA ligase is going to be the thing that fuses right there and fuses right there together. Ligate, to ligate something is to connect it. So this is an enzyme that's connecting DNA. And then histone proteins, if you remember, we have DNA wrapped around sets of 8 histone proteins, and these are called nucleosomes. So nucleosome 8 histone proteins plus DNA wrapped around it. One more time for DNA replication, this S phase. First, we have DNA unwinding by what enzyme? What enzyme is helping separate these double strands? This is called helicase, and this is taking place at replication forks or bubbles. So now we have these 2 exposed strands, and we know that we can only add from the 5 to 3 prime direction. Primers are going to come in. These are things that recognize that there's exposed nucleotides, and then DNA polymerase can come add from the 5 to 3 prime. On your leading strand, you're just going to add from 5 to 3, and it's nice and easy. On this lagging strand, you're going to make these Okazaki fragments, and then to ligate the Okazaki fragments, we'll use DNA ligase. After that, we're going to remake our chromosomes by wrapping them around these histone proteins. So this DNA plus histone proteins will help make nucleosomes, which are going to form our chromosomes. All right, hope that helps.